Hello students, today we are starting with the revision of chapter number 4, that is molecular basis of inheritance part 1. Okay, now I will revise all the topics one by one. Okay, in our lectures every topic I am going to revise one by one. So starting with the first topic today, we will start with the discovery of DNA. Okay, we have already studied the chapter. Now revision we are starting with. Okay, in 1869 Friedrich Mischer has discovered the DNA. Okay, discovery of DNA was done. What he has done, he has collected the white blood cells from the pus, okay, from the infected person, okay, the bandages of the infected person. Then he lysed the cells of the infected person, the pus of the lysed the cells of the from the pus and isolated the nuclei. He found a substance that is called as nuclein. Okay, so let us see this in Friedrich Mischer in 1869. What he has done, he isolated what he has called nuclein from the nuclei of the pus cell. This nuclein was shown to have acidic properties, hence it became nucleic acid. Okay, this nuclein that have been isolated shows acidic properties, hence it is called as nucleic acid. Nucleic acids are of two types, that is DNA and RNA. DNA means deoxyribose nucleic acid, RNA means ribose nucleic acid. Let us see in detail now what he has done. Friedrich Mischer have isolated. What he has done, he has done the experiment on white blood cells. For doing this experiment on white blood cells, what he has done, he collected pus from the infection. Okay, from the infection. For that, what he has done, for collection of pus, what he has done, he collected bandages from the local hospitals. Okay, he has collected the bandages from the local hospitals, and for removing the pus from the bandages, he used salt solution to wash off the pus from the bandages. Then he added weak alkaline solution to the cells, and because of that, the cells got broken down and nuclei got precipitated. So in this way, he separated the nuclei from the pus. Okay, and this nuclei, then it was named as nuclein, and this nuclein was having high phosphorus content and high acidic properties. Hence, it was named as nucleic acid. This nuclei was named as nucleic acid. Nuclein, since because of having the properties such as high phosphorus content and high acidic properties, it was named as nucleic acid. Nucleic acid are of two types, that is DNA and RNA. Okay, deoxyribose nucleic acid and ribose nucleic acid. Both are the genetic material. The next topic that we are going to study is the genetic material DNA. Okay, in 1900, the genetist knew that genes control the inheritance of traits. Everyone were knowing that genes control the inheritance of trait. You all have studied this in the third chapter, that is inheritance and variation. Genes are located on chromosomes. Okay. Chemically, chromosomes are mainly composed of DNA and proteins. Initially, most of the genetists thought that proteins are made up of they are large complex molecules and they store information for cell metabolism. But it was assumed that proteins caused the variations. What is happening? Proteins cause the variations. On the other hand, DNA was thought to be small, simple molecule whose composition varied little among the species. Over the time, these ideas about DNA were shown wrong. It was proved that DNA is the genetic material and not the proteins. Initially, the what was believed that proteins are genetic material and not DNA, but later, as the time has passed, it was concluded that DNA is the genetic material and not the protein. On the basis of certain experiments that were conducted from time to time, it was ultimately demonstrated that DNA carries genetic information and not the proteins. There are some direct evidence and some indirect evidence which proves DNA as a genetic material. Direct evidence definitely comes from the experiments that were done. The three experiments: Frederick Griffith experiment, Avery McLeod and McCarthy experiment, and Hershey and Chase experiment. These experiments were done one after the other. Frederick Griffith experiment was done in 1928. Then Avery McLeod McCarthy experiment was done in 1944. And Hershey and Chase experiment was done in 1952. Now, from our next lecture, we will revise all these three experiments in detail. Okay, one by one, we are going to revise all the topics in detail. Okay, Frederick Griffith experiment, then Avery McLeod McCarthy experiment, and then Hershey and Chase experiment. Okay, thank you for today. We will meet with our next revision lecture soon.